So it's a great pleasure to introduce Lars Steinmetz. He is a collaborator and a friend, and just as a very brief background, he went to Yale undergraduate, and he got his PhD at Stanford in Ron Davis's lab, and then has been really an innovator in genomics since then, and has managed to keep, he went to the EMBL and managed to keep his association with EMBL and Stanford, which, if you haven't noticed, they're not actually on the same campus. They're physically separated, and he commutes back and forth, but it really manages to keep a robust operation, and really has been a delight to collaborate with.
Thank you very much for the introduction, Bruce. It's a pleasure to be here, and I'm very grateful for the collaboration that we've started about a year and a half ago on DCM. I'll mention some of the work that we do in that space. Just to comment on the introduction, I often get asked what it's like to run labs on 2 continents. One advantage is that science goes on around the clock because there's a 9-hour time change between the two. So when one lab shifts off, the other lab comes on. And if you have collaborative projects across the 2 sites, then you can pass on work to the other location in the evening, and you get new results the next morning. That particularly works when you write manuscripts. So I'd like to tell you about, instead of just focusing on one aspect, I want to give you an idea about different projects in the lab that I feel most excited about currently. And they'll touch on a couple subjects. I'll tell you first about our use of yeast and the power of genomics in yeast to produce new natural products. And then I'll tell you about work that we do at the single-cell level, applying single-cell transcriptomic technologies to a few different biological systems where that really matters. And finally, I want to tell you some work that we do in the area of bio-device design in order to detect disease early. And in that context, I really want to start kind of with this slide as an introduction. I think we're really at an exciting time. Whole genome sequencing costs us now $850 per genome. And I foresee a time when every individual on this planet will be sequenced. Every individual will be sequenced at birth or even pre-birth in order to determine genetic variants in the genome. And then, of course, the question comes, what do you do when you identify risk factors for disease? And how can you offset the onset of symptoms that might occur later in life? And I want to kind of, to illustrate that, give you an idea of what you might find if you sequence an individual's healthy genome today. And so this is one individual that's still very healthy, but clearly has some disease genes in the genome, pathogenic variants that might lead to disease later in life. There are a variety of ones. RBM20, for example, that leads to dilated cardiomyopathy, autosomal dominant mutation. This individual is a carrier for that. You can also see variants for prostate cancer, and other types of cancers in this genome, and also a carrier for ApoE4, which is a predisposition gene for Alzheimer's, and a heterozygous carrier increases your risk by about 3-fold or so over population level, yielding a total risk of about 30% to get that. For RBM20, it's about 90% penetrant with an autosomal dominant mutation. You can kind of expand this list. This is currently 17 different predisposing variants. If you want to go a little broader and use all risk factors that are annotated in ClinGen, you can expand that list to about 60 variants for diseases. And you'll see similar things come up on here again, heart disease-related, Alzheimer's disease, and variety of different kind of cancers are coming up here on that list. So, I mean, I think that's what we're going to face as we sequence genomes. And how do we determine, when it's critical to intervene with a therapeutic approach? We need measures to measure progression of disease at a molecular level, and we need really ways in which we can interfere with that progression, and so focus much more on the treatment of individuals while they're still healthy than when they've developed disease. So at the Stanford Genome Technology Center, which I now co-direct, and I've been associated with the center for 21 years, we focus on development of novel technologies that will transform biology and biological investigation. We both develop technologies that improve our ability to study natural biological processes, genome biology as a whole, but also technologies that can distinguish between health and disease state, and technologies that allows us to cut the costs of healthcare and enable better and more accurate diagnostics ultimately. So in my talk today, I'll touch on areas of research in all of these different diverse fields, and I will start with a project on natural products. So here we really take advantage of yeast. Simple eukaryotic genome, the first one sequenced actually right around the time when I started my PhD. And so a lot of my PhD work was focused on yeast functional genomics at the time. We have started a project where we wish to tap into new natural product diversity that's out there in nature that previously couldn't be tapped into because 90% of the organisms that produce natural products cannot be cultivated in the laboratory. And in addition to that, only about 10% of natural product encoding enzymes are expressed for those organisms that you can culture in the lab under these laboratory growth conditions. So really, we're missing the vast majority of natural product diversity out there and can't test that for bioactivity, both in terms of understanding basic biology, but also for therapeutic applications and the number of drugs that are based on natural products. In fact, 2/3 of all anti-cancer drugs and antifungal drugs are based on natural products. So potentially there's a lot more out there that we'd like to get our hands on. So with today's technologies, you can sequence genomes, though, of organisms that you can't cultivate in the lab. We can do genome sequences even of single cells. So we can get at their genetic composition. We can't grow these organisms to isolate natural products. But bioinformatically, we can mine the databases of such sequence information, predict putative natural product encoding enzymes, synthesize them from oligonucleotide synthesis or gene synthesis from scratch, and then put them in organisms that we can cultivate in the lab. So that's the project that we've undertaken, and we're using yeast, Saccharomyces cerevisiae, as a powerhouse to produce these new natural products. And so we're part of a Genomes to Natural Products network that's funded by NIGMS, and they're supporting research project teams in different areas. So we're the fungal group, looking at natural products from other fungal species. There's also a different group working on bacteria and another one working on plants. But the goal of this network is to develop new tools and technologies that will enable one to do this type of research where you predict the enzymes bioinformatically and then produce them in an unnatural host organism. So the way we would do this for S. cerevisiae, I already mentioned kind of the pipeline. We take fungal sequence information. We use bioinformatic tools to predict biosynthetic gene clusters from those genome sequences, and we are aided here by the fact that natural product encoding enzymes occur in clusters in genomes. So often you have these, if you have one biosynthetically encoding enzyme, you have the other enzymes that are part of that same synthesis pathway right next to one another in the genome. So we can use bioinformatic tools to look for similarity to known natural product encoding enzymes, polyketides, terpenes, or non-ribosomal peptide synthetases, and then predict neighboring genes bioinformatically, then synthesize them through gene synthesis technologies, clone them into plasmid vectors that then contain these genes. Typically, it's 6 to 7 genes per cluster. We transform them into yeast S. cerevisiae, let them be expressed, and then we use mass spec in order to see whether we can detect a new mass spec peak from these cultures that might be the final natural product, but not necessarily. Could also be an intermediate along the path of the synthesis process, but it allows us to then identify enzymes and sequences that might encode new entities. And so we've done this. We're now close to 4 years into this project already, and we've processed 100 enzyme clusters so far and taken them to this pipeline. It's about 6 weeks from beginning to end, and the final step is characterization by mass spec of new compounds. And you can see that we succeeded for about half of the enzyme clusters to take them through the entire pipeline. We have some failures along the way, and that's mainly due to incomplete prediction bioinformatically of where the splice sites occur in these enzymes. Because in related fungi, there's a very active splicing, while in S. cerevisiae, there's almost no splicing, which means that we're resynthesizing the enzymes without the introns, and we have to predict exactly where the exon-exon boundaries are, and there's some failures here. But of the things that we reach, where we get measurements all the way at the end, we succeed in detecting a new entity in about half of those cases. And so these distribute into polyketide synthetases or terpenes that we've processed with this. We're not really focused in this grant on studying bioactivity of any of these entities. We're very interested in that question, but we're explicitly precluded from looking at that so far with this particular grant. But that's a direction we want to take going further with these. I want to point out one limitation of this technology so far, and it's mainly the cost of DNA synthesis is still way too high. So with next generation sequencing technologies, we saw a dramatic drop, 10 to the 5-fold or 10 to the 6-fold in terms of sequencing costs. With synthesis, we haven't seen that revolution so far. So we still pay about 10 cents per base in order to synthesize these enzyme clusters. And you can see here in terms of our entire costs for making these plasmid libraries, most of it goes into the gene synthesis companies from which we order the DNA. And so we can't really drop the costs much further unless we improve DNA synthesis capabilities or work with other suppliers that can really push those boundaries to a lower level. I mentioned gene expression as being a major limitation for our success in getting these clusters to work. And so that brings me to the next topic of what I want to touch on in my seminar today, and that's transcription and ultimately single-cell transcriptomic methods. And my lab, when I started it in 2003, we had a focus on defining transcriptional complexity across the genome. And in kind of 3 slides, I want to give you a little bit of a background as to what got us to look at single-cell transcriptomics with these methods. So at the time, we used microarray technologies. In fact, we worked with Affymetrix to design the first tiling array to systematically measure transcriptional activity across the yeast genome. And that led us to discover that most of the DNA in the genome is transcribed into RNA. It was called pervasive transcription, a phenomenon that's been seen in every organism that's been studied so far. You get transcription often on both strands of the genome, making sense-antisense pairs, but collectively, even under rich growth conditions, 90% of the yeast genome is transcribed into RNA molecules. And you see similar numbers if you look at the human genome as well. We then went on to ask, how is this transcription actually conferred? What gives rise to it? So we looked at promoter activity, and we realized that most promoters in the genome fire in a bidirectional orientation. So not like the classical textbook understanding where you have a promoter and transcription in one direction, making a protein coding gene, but in fact, I think every promoter fires bidirectionally, and it makes a protein coding RNA in one orientation and a non-coding RNA in the other orientation. And the reason we never picked those up before is that some of those RNAs are very unstable and they're rapidly degraded already in the nucleus. But you could see this activity of bidirectional transcription. And then if bidirectional transcription occurs in such a fashion that it generates a non-coding RNA that runs all the way through a protein coding gene on the opposite strand and overlaps to the promoter region, then you can get antisense regulation of that promoter. And we've shown that antisense transcription can repress transcriptional initiation from these promoters. And that then suggests that you could get spreading of regulatory signals across the genome through these long non-coding RNAs that are being generated, where you're controlling this promoter really and controlling this open reading frame, but indirectly you're also controlling the upstream open reading frame at the same time due to the non-coding RNA that interlinks these. We further went on to look at transcriptional complexity that's being generated at all of these positions. So not only is every nucleotide transcribed into an RNA, but every position is also transcribed into different isoforms of RNAs that differ in terms of where they start and stop throughout the genome. And I'll show you some data on that. It was really staggering, and we were very surprised when we saw that complexity. And that can mean even for a simple eukaryotic like yeast, where you don't have any splicing or alternative splicing occurring, you can diversify the proteome by only partially transcribing open reading frames. So if you generate one isoform that initiates here, you can make another isoform that generates a truncated version of that protein, and in some cases there's a signal peptide here that gets included or excluded on the open reading frame, generating an extracellular version of the protein and a cytoplasmic version of that same protein. And it can also has the potential to encode new short peptides that are hidden open reading frames in the genome. We developed a number of technologies over the years to look at transcription, both transcriptional heterogeneity, but also chromatin heterogeneity, and to multiplex these systems. I won't say more about that, but I want to show you some data from this transcriptional heterogeneity that's being generated in this genome. And this is a snapshot of just one random region of the yeast genome. You have protein coding elements here that are encoded in the Watson strand orientation on the top, and then others encoded on the Crick strand orientation on the bottom. And what you can see here are tracks from a method that we call TIFF-seq, transcript isoform sequencing, where we sequence the ends of full-length cDNAs. And we do this by generating a full-length cDNA that we circularize to bring the 5' and 3' ends together. We purify just the sequences around the junction site and use next-generation sequencing to sequence across the junction. That gives us single nucleotide resolution on the 5' end and 3' end of one and the same RNA molecule that originated from. And this can be done in bulk across an entire population of cells. And so in a single snapshot, then you get reads where every line here is one read with a unique kind of start site and stop site across the genome. And you can see that there's a huge amount of diversity that's being generated from just one culture of clonal population of yeast cells grown under rich media conditions. You can see things like bicistronic RNA messages here, where this here is an RNA that spans these two adjacent open reading frames. You get lots of variation on overlaps, where one transcript overlaps the 5' end of a downstream message, and lots of variation in the UTR elements, and even this internal transcription initiation inside the open reading frame that has the potential to generate truncated protein messages and therefore diversify the function of the proteome. And we were very surprised by the number. Yeast has 6,000 open reading frames. Under just rich growth condition, we already detect 1.8 million unique transcript isoforms that are being generated from this genome. That's on average about 26 major transcript isoforms per protein coding gene across this genome. And again, this is in the absence of splicing. So I think if you think about a mammalian genome, you'd have that diversity plus splicing diversity added on top of that to diversify the transcriptome. And it's not just that there's one major isoform and everything else is minor, if you consider the isoform diversity for a gene, but you need to consider 10 isoforms to explain 80% of the expression of a gene. So there's a lot of diversity in there that we previously couldn't really measure with microarray technologies, where we would only, for example, have one probe per message, or with RNA sequencing, where standard RNA sequencing, where we fragment the genome, because you can't see isoform complexity in these data. So that data really led us then to think about single cell resolution, because to me, one interesting implication of these data is that this could mean that there's a huge amount of diversity between individual cells. We had known before from other data that in yeast there's an average of one mRNA isoform per gene per cell. So if there's only one isoform that's being generated for each gene in each individual cell, but you have about 30 possibilities to choose from across 6,000 genes, then probably no two cells in the population are making the exact same transcriptome. They're choosing different combinations of that, and that could then really diversify a population of clonal cells. And I think that's important for fitness for clonal populations, because it could mean that a single cell is making the right transcriptome to survive in environmental stress conditions, so that the genome can then be passed on, and the genome has the ability to regenerate all that heterogeneity upon clonal expansion of those cells. And so we wanted to test that hypothesis and really measure transcriptional heterogeneity at an individual cell resolution. And at the time, it wasn't possible to do this in yeast, because yeast has too few RNAs per cell, only about 60 to 100,000. But it's possible to do that in mammalian cells, because mammalian cells have 300,000 mRNAs in a single cell. And so we could study this in mammalian embryonic stem cells. And so Lars Velten in the lab did a study where he measured 3' end isoform diversity in these cells, and could show that between individual cells, you have different isoforms that are being expressed, but also that in mammalian cells, you're making multiple copies of the same gene, multiple mRNAs from the same gene in an individual cell. And it was always the same isoform that tended to be made repeatedly. So there's something about the isoform choice being encoded probably in the chromatin or epigenetically in an individual cell, but there is that heterogeneity and diversity that exists. We've also done a study in thymic tissues where that plays a role to make sure that no T cells escape into the bloodstream that might recognize self-antigens. But I want to tell you a little bit more about a study that we did most recently, where we looked at the hematopoietic system in humans. And so we isolated bone marrow samples from healthy bone marrow donors, and we applied single-cell RNA sequencing to these cells in order to reconstruct the lineage differentiation tree of early hematopoiesis. The classical model, as you might remember from textbooks, et cetera, is one where you have multipotent progenitor populations that then differentiate into bipotent progenitors and ultimately unipotent progenitors, and there's binary branching decisions that happen along the way. We think that model doesn't fit the actual data, and I'll show you some data that, and this is a graphical representation of what our data suggests is happening in human hematopoiesis at steady state, and that there's rather these stem cells kind of in the center that then linearly and gradually differentiate to their final states, and you don't have really these binary branching decisions that occur along the tree. And this is a massive amount of regeneration that happens in a healthy individual every day. So up to 1 trillion cells, blood and immune cells get regenerated through this process in a healthy individual in a daily fashion. And so the approach that we set up to do this also was aimed at overcoming one technical limitation of single-cell methods, and that's typically that if you do a transcriptome on a single cell, you can no longer do any functional studies on that cell because you've obviously killed the cell to isolate all the RNA. So any determination of what fate that cell would have acquired later if you would have let it differentiate down the road, you cannot capture. But we went around that process through a method that we call index omics, where we use 11 FACS surface markers that we can measure, but we don't use those surface markers to do any sorting. But we can register the abundance of expression for 11 different surface markers, sort cells, then about 1,000 bone marrow cells that are CD34 positive into different wells of a 96-well plate. And for each one, we register exactly the expression level of each of the 11 surface markers. We can sort 2 sets of plates. One set of plates we can process for single-cell RNA sequencing to isolate the RNA and look at the transcriptome. The other sets of plates, we do single-cell cultures, both in vitro culture differentiation, but also xenoplant kind of transplantations to determine cell fates that these cells would have acquired. We can match these 2 data sets back to one another because we have the 11 surface markers that we've measured on the individual cells. So you can then group these together, and we did about 1,500 cells, individual cells from 2 donors that were processed with these 2 methods, and then the data were merged together. Now I want to show you the data on the transcriptomics that we found surprising. So we took populations of cells that classically, and this is actually the classical tree model of hematopoiesis, so we took cell populations that should in principle belong to this category. There are 4 major cell types that are defined at that stage. If you take those cells and you do clustering of them based on their transcriptome diversity, you get a graph like this. We get 16 different clusters that come out, not just these 4 clusters, so some substratifications, but more or less the clustering and the genes that they express corresponds to end cell states that we finally expect to get out from the differentiation tree. The real surprising thing was when we started to look upstream, and we looked at cell populations that classically were in this part of the tree, where the model predicts 3 distinct cell states. And when we do clustering, we don't get any clusters that emerge from the transcriptomic data. You can see it here. It looks like one kind of uniform cluster with some slight variations, but clearly not 3 distinct cell states that emerge from these data. So we then wanted to ask, how does it happen then that you get from an undifferentiated kind of mixture of cells to distinct cell states as you move through this tree? And for that, we developed an algorithm, a bioinformatic approach to determine similarity of transcription of cells in that undifferentiated compartment to one of the final end cell states. And we do this by plotting the end cell states on the periphery of a circle, and then every dot here is the transcriptome of a single cell. We then place that cell and its transcriptome based on the similarity to one of the end states. So we can both measure direction of priming and the degree of priming with that method. If you're staying multipotent and you're one of the stem cells, you should be in the center because you haven't adopted a transcription signature of one of the final states. As you start to adopt a signature of one of the final states, you move out kind of along into the direction of that final state. And you can see right away that this looks like more like a star-like model where fanning is coming out directly out of the center, and we don't have these binary branching decisions where you first migrate into one common direction and then eventually you split as you proceed down that tree. In addition, there are no distinct cell states. It's very gradual. There's cells at all distinct states here, so they're gradually acquiring a transcriptional pattern towards one of the end cell states. So that really led us to this conclusion that the classical model of hematopoiesis doesn't really fit the transcriptomic patterns that we derive from bone marrow cells that are isolated from patients and then directly processed post in vivo isolation. We think this model here, without binary branching and with very early priming and with continuous transitions, much more like a Waddington-like landscape of differentiation, fits the data much better than a tree-like model of differentiation does. So I want to also tell you about another study that we're doing. This one is still unpublished, in collaboration with Mark Davis, where we're looking at T cells in particular and how T cells respond post-infection. And we're interested here in asking the question of how a T cell response is controlled at single cell level. And what happens when T cells see a pathogen is that those cells that have a TCR, a T cell receptor that recognizes an antigen, will clonally expand in the population. They get activated, and the activation leads to an expansion of those T cells with the same TCR sequence that they have in order to fight the pathogen. Eventually, when the pathogen is overcome, you have a recovery state, and you end up with a collection of memory T cells where there's a slight reduction of that clonal expansion. Some of the clonally expanded cells undergo apoptosis, but you still maintain a repertoire of diverse TCR clones post-infection. And how this is controlled, so you experience clonal expansion of individual cells, but you don't clearly want to overtake the entire population, because then when you reach a memory state post-infection, you don't maintain any TCR diversity anymore. So we wanted a method that allows us to look specifically at clones and to look at cells that have expanded a lot in the population of activated T cells versus other T cells that didn't expand as dramatically, to see if there are biological cues that these cells acquire that limit their further expansion and control this process. And so we set up a project together with Mark Davis's group where we worked with 5 different patients that had infections for MTB, but are no longer chronically infected or acutely affected by MTB, but they have memory T cells in circulation from that pathogen. We can isolate PBMCs for that, and then we do a treatment with MTP lysate for 12 hours. That's not enough to induce proliferation of these cells, but it's enough to activate them so that we can identify T cells that respond to MTP antigens. And then we can generate a sequencing library for these cells. We process 3,000 T cells for that, and then we do a targeted amplification of just the T cell receptor cDNA in those libraries in order to determine whether they belong to a clone or whether they're singletons in the data set. And we can match clones based on similarity in the TCRs, because essentially there's a 10 to the 16 diversity of TCRs in the population. There's a very almost non-existent chance that you get that same TCR 2 times in this population of 3,000 cells. So we group then cells based on their T cell receptor into cells that belong to clones that are all similar in terms of the TCRs, and others that are singlets in our data set. So from 3,000, we did selection of cells, because only a fraction of those are clonal and others are non-clonal. We selected about 1,000 cells in total, and then did in-depth transcriptomics on that population to define the transcriptome. And we sequenced at a depth of about 2,500 to 3,000 transcripts per individual cell. And so then we start to look at these data. And so our data allows us to also define 2 distinct populations of T cells. We have conventional T cells and unconventional T cells. The unconventional T cells are made in natural killer cells in that population. So we first looked at the global diversity of these cell types. And we can see that the conventional and the unconventional cells clearly separate based on this T-SNE map, which measures similarity in transcriptional profile data. But we don't get really any good separation between the two distinct unconventional cell types. So NKT and mate cells, to us, look the same, essentially, based on their transcriptome. But you see clear distinction between conventional and unconventional cells, and a huge amount of transcriptional diversity across activated conventional T cells. And so those are mainly the clonally expanded cell populations, and so we were interested in looking at that in more depth. So we did a separate PCA analysis on the transcriptional heterogeneity of the clonally activated T cell population. You can see here that there is clear separation. There are two principal components of these data. And if you now plot for each individual cell, whether it belongs to a clone that has expanded a lot or it belongs to a clone that hasn't expanded, which I'm plotting you here based on both color, gradient, and size of the dot, you can see there's a clear separation along principal component 1, which is the main axis of differential transcription in these data. Cells that belong to large clones fall on this side of the PCA plot, while cells that are singlets in the data fall along that side. So obviously, there's a clear transcriptional signature that correlates with clone size, and we were interested in determining what genes are differentially expressed among these different clone size groups. And so we looked at that in more detail. This is just another representation of what I just showed you on the previous slide. PC1 mainly correlates with clone size, as I showed you. And then we looked at cell proliferation genes. And interestingly, we saw a clear anti-correlation between the clone size and the number and the expression of cell proliferation genes in the data. And I was surprised because this was counterintuitive. I thought cells that belong to large clones, they clearly have expanded a lot. They've proliferated a lot. I would have expected high expression of cell proliferation genes in large clones. But it's exactly the opposite. Large clones have downregulated cell proliferation, and small clones have high cell proliferation capability, which fits to the idea that in a large clone population, at some point you need to limit further expansion, and you downregulate cell proliferation genes. The gene that comes up at the first list in terms of determining that this anti-correlated pattern is AL2RA. And AL2 is a factor that promotes proliferation of cells, and we see that the receptor for AL2 is downregulated in clones that have expanded. So they're no longer sensitive to the AL2RA receptor, and we've verified this is from RNA-seq data. We also see that when we look at protein data, and that leads us to this model where we think that when you see an infection, you get expansion of T cells into clones, and then eventually, after a certain amount of expansion and proliferation has happened for a clone, it downregulates the AL2RA receptor, limiting further proliferation, and then maintaining a repertoire of the TCRs for further pathogens or if that antigen is seen again in the future. I showed you data that focused on short-read RNA sequencing at single-cell resolution. We've also thought a lot recently about doing long-read RNA sequencing, in particular for cases, for biological contexts where long-read RNA sequencing provides more information or information that's necessary to understand the biological process. And this is the case in dilated cardiomyopathy, a project that we started also in discussions with Bruce Conklin's lab. And we're focusing in particular on dilated cardiomyopathy mediated by a gene called RBM20. This gene leads to severe forms of dilated cardiomyopathy. It's autosomal dominant. A lot of the mutations cluster in an arginine serine-rich region of the protein sequence called the RS domain, and you can see here clustering of all or many of the known mutations along that region. They're pretty much immediately adjacent to one another. And we got particularly interested in a new mutation, P633L, that we've identified in a family with this disease. And so what was known already before is that RBM20 mutations lead to missplicing of key cardiac transcripts, including titin, the ryanodine receptor, CAMK2D, genes involved in contraction strength and frequencies of contractions. of these cells. And what is interesting to me is that not only do mutations in RBM20 lead to these splicing deficiencies, but also the levels of RBM20 expression in patients matters too, and they have very similar effects. Low levels of RBM20 functions very similar in terms of the splicing defect to point mutations in that gene, and that's what's shown here with the red and the black comparison. And that's true for a lot of these different target genes. So we set up a program in order to study individuals with this disease and to get a better idea, and particularly with this focus of this new mutation. And we thought of different things that we could do with that, a lot of it inspired by what Bruce has already pioneered here in the system, generate iPS-derived cardiomyocytes that we can use CRISPR editing on to fix patient mutations back to wild type, or we work with isogenic lines to introduce a variety of RBM mutations in that background, and then study these cells both with transcriptomic methods, but also phenotyping technologies. Develop a mouse model for this P633L mutation by using CRISPR technologies to engineer this into mice, and we are working on that. We have an RBM20 knockout mouse where we've mutated the RS domain entirely so far, and in collaboration with a company looking at a pig model for RBM20 loss of function. We also want to develop devices taking some of the technologies from the Genome Center to detect this disease earlier, and ultimately think about screening technologies and other therapeutic strategies. So we've done iPS-derived cardiomyocyte differentiation. We get cells that are beating in the dish at a little bit less than 60 beats per minute, and from that we can do RNA sequencing studies. The RNA sequencing studies that we want to do include full-length RNA sequencing because this is a splicing defect. RBM20 is a splicing regulator, and it missplices the target genes. And we want to know exactly which isoforms are produced as a result of dysfunction in this gene. With short-read RNA sequencing data, you only see abundance measures of different exons within the open reading frame, but if there are two exons that are misspliced, you don't know the combination in which they're being generated. So we work with Oxford Nanopore sequencing technology where we can generate full-length cDNA sequence reads for about 4 million transcripts on a single MinION chip. And we're getting a base pair output in terms of sequencing that's the same as on an Illumina MiSeq machine. So the technology looks very promising. I'll show you some data that we've collected so far. So here's a length distribution on a bioanalyzer trace of the cDNA that we're putting in the instrument. And you can see there are maximum of about 10 kb fragments in here with a span, all the way to about 500 or so. This is a distribution of the actual read lengths that we're getting out from the sequencer, and that distribution looks very comparable. We're also detecting fragments here in the 10 kb range. In fact, we can sequence all the way to 100 kb fragments with this method, and the distributions kind of are comparable with that. The error rates are very high in terms of base call error rates. They lie at about 15%, but for exon identification, it's good enough. So we can identify the exons that are included in these cDNAs. It also looks like it's quantitative. If you do a comparison to spike in material, you get a linear proportion, also comparison of short read to long read. Sequencing quantification of this method also looks very linear. So not only can you quantify, but you get, in this case, full-length isoform information. And that's what it looks like. I'll just show you some examples. Here's a gene for which there are 5 different isoforms that are annotated. The annotation here is shown in red, and these are the actual sequence reads that we're getting out. So we're detecting the abundance of diverse isoforms for that particular gene that are present in the sample. And of course, we can now do comparisons of mutant versus wild type to look at splicing changes that occur in these samples. So I'll show you one gene here where there's a novel isoform that's even detected here that isn't in the annotation. You can see slight enrichment of that in the RBM20 wild type. The mutant has slight depletion of that, but different abundance of this exon, for example, isoform compared to that one. So we're mining these data now. This is still work in progress, but with this technology, we can kind of look at that resolution. Ultimately, we'd like to drive this down to single cell resolution. We have a project in collaboration with a number of other labs that's funded by the Chan Zuckerberg initiative to try to do this at single-cell resolution in heart, in mice and in human samples eventually. But this is a pilot project funded for a year to kind of get started with technologies along these lines. But hopefully we'll have some data here. So in the last couple of minutes, I want to show you some work that we do with sensor technologies. I mentioned all of the molecular biology that we do. We also want to kind of think about detection of diseases early and to think about wearable technologies that would enable us to do that. And this is a depiction of kind of what I think how medicine in the future could look like. Combination of genetic information that I already showed you on the previous slide with integrated omic profiling that can be done periodically over time. But importantly, wearable technologies that I think will have a large impact on this in the future. And many of you are wearing activity types of tracker technologies. What I'm thinking about here is more biochemical sensors that would be able to register the expression of individual genes in real time, in individual. So you could detect kind of onset of disease and patterns there. And so I'm part of a study that does this with an activity tracker over time, this Mike Snyder study that he rolled out to 100 individuals. I'm now into the study about 3 years or so. I'll show you just one data from a Fitbit tracking device that I was wearing here from a day back then, so you can read kind of pulse patterns out over time. This was actually me traveling to a scientific retreat in Germany. So while I was going on the Autobahn, kind of pulse rates spiked up. And then when I arrived at the meeting, it kind of declined. Then the scientific talk set in, and it kind of went really down. And then finally, there was drinks and dinner, so that got the pulse rate up again. You can see interesting things here, but what you really want, I think, is the biochemistry and drive it to that level. So one of the senior scientists at the Genome Center has worked on a sweat sensor that he developed where he can measure 5 different entities routinely. So he works on a flexible integrated circuit that you can wrap around your wrist that has these sensors integrated in a collaboration with a lab in Berkeley that can measure glucose, lactate, sodium, potassium, and body temperature in real time. And he's developed a sensor, different types of sensors on that surface, enzymatic as well as electrical sensing technologies that are on an integrated circuit with a Bluetooth transmitting device that can send this information in real time to a smartphone app where you can then read out levels of these different things over time. And so they did a study. They did this in Berkeley rather than in Stanford, I think, because I guess in Berkeley nobody says anything when somebody runs around the campus with an integrated circuit on their forehead. But they got participants to volunteer, 4 people. 2 of them got regular water intake to be rehydrated, and the other people did exercise without water intake, and they measured the response, of these different things over time, and they saw that with sodium and potassium, if you get water intake, you get an initial kind of reset of the level, but eventually it kind of stabilizes. While if you don't get water intake, you have another spike that happens after about 80 minutes, which corresponds to the onset of dehydration. So you can use this technology to measure dehydration status. Ultimately, you want to apply this more towards medical applications. So Sam has now set up his own lab at UCLA to follow this in more detail and apply it to cystic fibrosis samples in particular, but he's kind of pursuing that on his own. I want to tell you about one technology that we work in the lab, where a postdoc is working with me on using magnetic levitation in order to characterize cells and cell behavior. And this kind of was inspired by an experiment that Andre Geim carried out in 2000, actually, for which he got the Nobel Prize. He took a live frog, and levitated in a 16 Tesla magnetic field. And you get the frog levitating there. He went on to do greater things, so he got a Nobel Prize for the discovery of graphene 10 years later. But it was thought at the time that he did this experiment that you couldn't levitate anything smaller than 20 microns in size, and so this technology wouldn't work for characterization of single cells. So together with Utken Demirci's lab, who actually started at Stanford at a similar time than I did, he developed a device where he took 2 magnets and he oriented them very close to one another with just 1 millimeter spacing from each other. And in this milieu, which experiences a very strong magnetic field, you can insert a capillary that contains cells that are immersed in a paramagnetic fluid, and we just use paramagnetic fluid because it amplifies the magnetic field strength, but it's a regular MRI contrast reagent, Gadavis, that everybody gets injected with for an MRI procedure. So we're thinking and hoping that this is non-toxic to cells. We then put, when you put cells in that space, gravity pulls the cell down, the magnetic buoyancy force pushes the cell up, and it equilibrates after about 20 minutes or so. And the height of levitation corresponds to the density that the cell has. So if you work out the equations of this process, it mainly corresponds to density. So you have a technology that doesn't need labels, no centrifugation, on a small portable device where you can separate cells based on density. You can apply that to different types of blood cells. I'll show you a video here. Red blood cells initially dispersed, bottom magnet and top magnet, and then here within minutes, kind of they eventually form a uniform band in the center that then corresponds to the density that they have. So you can apply that to sort cells of different populations. So you can, red cells and white cells, for example, have different densities, so they levitate at different heights. In this, and you can see that here in an experiment on top. Also, activated neutrophils versus unactivated neutrophils have different densities, so they could be separated. And also, as an example, sickle red blood cells versus healthy red blood cells also have different densities with this technology. One application that Ghost has been exploring is whether she can use then this technology to sort out cells of different densities from blood. And one application would be to sort out circulating tumor cells out of the bloodstream, because tumor cells have lower density than blood cells have. They're larger cells. And so they levitate at a higher height than the red blood cell population is. And so she's been exploring that, and this is a process that happens when during the epithelial to mesenchymal transition as these cells migrate into the bloodstream. And there's no real good technology out there to get these cell types. A lot of the technologies use EpCAM surface antibodies to pull these cells out, but during the EMT transition, EpCAM is actually downregulated. And so some of the cells that are undergoing that transition no longer express EpCAM, and you don't capture them with that technology. So could we use this kind of label-free method to just sort cells based on density? And then we have a microfluidic device that pushes the sample through and a splitter at the end, where then the red and white blood cells go to the bottom channel, and the circulating tumor cells will go to the top channel. And so this is in principle, very similar to this kind of in concept, but it happens on a very small kind of portable device. So you just take two stationary magnets with some plastics. Actually, here's an amplification of what this looks like. And you need just a simple microscope that can image cells, that's good enough to image cells, that can look at that space between the two magnet surfaces. It costs about $1 to manufacture the hardware components for this technology, and the reagents are negligible in cost. So this paramagnetic fluid doesn't really, not a significant expense at all. So she's done an experiment where she has taken a sample with circulating tumor cells, spiked into a blood sample, and these tumor cells are fluorescently labeled. So you could see them in green here, and she's doing a flow of about 2 ml per hour of blood through that channel. And you can see these tumor cells migrating to the top channel as they're flowing through, and the magnetic buoyancy force is pushing them up, and blood cells go to the bottom. Occasionally, there's a green dot going to the bottom. These are dead tumor cells. When cells die, they become denser, and they go to the bottom. So it's also a device to separate live versus dead cell populations. When we collect these cells, we can do transcriptomics or any genomic characterization on them, and that's what we would like to do. So we can also measure responses of cells to different drugs. So you can take chemotherapy on these cells and see whether they're dying in real time. So here's an experiment with breast cancer cell lines that have been exposed to hydrochloric acid, and then over time here in minutes, you can see individual cells, even though it's a clonal population, dying at different stages, and they're pretty much dropping in the magnetic field. So you can then imagine setting up a device where you isolate cells from a patient, you then put them into another chamber, magnetic field chamber, with different chemotherapy substances. You see which ones the cells respond to until you have a combination cocktail that would be effective on all the cells that have been isolated. And potentially all could be done while the patient is still in the doctor's office, because it's a fairly rapid and inexpensive to do, and can be done on a simple kind of portable device. You could similarly apply that to antibiotic susceptibility testing, bacterial levitate 2. And so here's an experiment with E. coli exposed to different antibiotics, and you can clearly see differences. Here, actually, two populations emerge. This drug induces filamentous growth, filamentation in some cells, and in other cells of the population, they die. This drug just induces cell death, and it's very similar to classical OD measures that you get. Even yeast cells, at different stages of the cell cycle, have different densities. And so when you levitate a cell population that's still growing and dividing, you get these cells levitating at different heights depending on cell cycle state. And the device is small. In principle, you can couple it to an iPhone camera that has a small attached lens that is able to measure the cells that come out. And you can use computer algorithms to quantitate how many cells there are of a particular type in these devices. So with that, really, I want to finish. Want to thank all the people in the Stanford Genome Technology Center that have contributed to the development of these novel technologies, some of which I could present, and also the people in my labs, both in Heidelberg on the left and in Stanford on the right, and in particular collaborators, Bruce Conklin, Utke Demerci for the collaborations on the magnetic levitation, Wolfgang Huber and Marieke Essers, and Andreas Trump for a collaboration on the hematopoiesis system that I presented, Mark Davis for the work on the MTB clonal expansion. And I want to thank you all for your attention.
First question gets to be levitated.
Mm.
Again, you see the small ones becoming larger because they're proliferating out upregulation. It would become a larger subset and vice versa?
I would think so. I don't know if there's communication between the different clones towards one another. That I don't know, but I do think so. There's dynamics there where the larger clones get kind of experience upregulation of senescent markers and downregulation of proliferation genes, and the smaller clones, they don't have that, so they're actively proliferating. And so you have an ability kind of to maintain a TCR diversity even in the face of a strong pathogen.
I had a question about your the sort of TIFC and the variability results. So you mentioned that it might be the cell-to-cell variability might be adaptive in some way. Have you tested whether the heterogeneity increases under stress conditions?
That's a good question. So we've done galactose versus glucose growth conditions only. We haven't done any stress conditions so far. So I don't know if the diversity as a whole increases, but my expectation would be that the subset of genes that experience diversity and heterogeneity changes between the two conditions. I would still think you get like 1.8 million distinct isoforms, but a different subset of genes are expressed, and a different subset of genes experience large isoform heterogeneity, and that would be kind of important. So in order to address that, what we've tried to do is set up a system where we trap individual cells in microcolonies, so in alginate beads, individual cells, and we can then grow them up with time. And so we don't have to do single-cell transcriptomics, but can do transcriptomics on clonal populations at different times of division. So we get cells that have only divided a few times to cells that have divided many times. You anticipate that this heterogeneity initially is doesn't exist, or there's more uniformity in the population, but heterogeneity will increase as this population expands. Because clearly, when we look at the full population, we get the huge amount of heterogeneity, but at a single cell level, we don't see that, right? And the question for us was also, at which stage is this fix on one isoform released on the system, and at which point you regenerate heterogeneity? And so with populations like that, you could do functional analysis too if you identify these cells having particular characteristics or reduced isoform heterogeneity. You could test for fitness effects under stress conditions and other things.
Differentiation models of a pedigree, that is a cell division pedigree, where you can actually look at sister cells.
Right, yes. You mean for the yeast experiments?
Well,
yeah, or any of them, yeah. They would apply to any. We haven't done any investigations of proliferation in vitro where we would separate mother-daughter cells, for example, then do transcriptomics on that. I've been trying to convince a student to take that on. It's a lot of work because you got to sit there and visualize the cells and then separate right after division and then, get them into separate wells. But I think that would be a good experiment to do, also to ask kind of how much of this lock-in of one isoform is inherited. Like similar to the question that I just talked about, the one that we came up with, the alginate bead, is one where less labor is required from the personnel, but the other one would be equally interesting and probably more direct.
It's really interesting to see this continuous differentiation instead of a very discrete one.
Mm-hmm.
And I was wondering, have you confirmed that these cells that are sort of intermediate are those same cells that are then progressing to transit-terminal states?
So we've done this kind of in vitro differentiation of the cells to look at which fates they acquire. And we have taken cells at different states, as long as they've already kind of on our signature, it looks like they've already acquired lineage potential. We've taken cells at different positions there and then cultured them and shown that indeed all these cells reach the final state. And they don't reach other cell states, so they're not like skipping into other branches. So those experiments exist. But maybe also along the question kind of previously, I think what is really lacking in the system is a genetic tracking tool that would be able to track, obviously cell differentiation in vivo, but you can't do that in human because you'd have to genetically modify it. So there are a number of technologies that use barcodes and mutatable barcodes, et cetera, over time where you can do barcode tracing experiments. There have been papers that have done this in mice, but there's limitations on those experiments, and a lot of that is confounded by the half-life of the cell population, from which, because they come from different stem cell progenitors. If a cell is very short-lived, it must have arisen from stem cells that have differentiated late in the process, while very long-lived cells can have differentiated from populations throughout the entire spectrum of the hematopoietic kind of stem cell continuum. So just looking at similarity of barcodes isn't really the right tool to do that. So I think this question is still outstanding, but it would be good to have genetic tracing technologies to validate some of these methods, because also the in vivo differentiation processes, I mean, that's what people have done, in the past too, and that's taking things out of context. That's why we like the system where we just take the cells from bone marrow directly and map the tree based on just the similarity of the transcriptome, which at that stage should still be unperturbed.
Okay.
Yeah.
One more question, and then there'll be a lunch with training.
Along the lines of the last question, how do you square your data with other experiments that have led to the current model of bifurcations based maybe on lineage tracing experiments?
How do we kind of think about that or resolve these issues? Yeah, I think it's a very good question. So, I mean, there have been a number of papers, or one in particular that has come out after we published ours, putting this model into direct question through a lineage tracing experiment in mice. But reanalysis of their data shows that they didn't consider kind of cell half-life states, and it's confounded by what I just kind of explained in terms of where the cells derive from. So I think they reached a conclusion that they can't really reach yet, I think, with the data that they generated. And it still leaves open the possibility for all models from their data, if it's analyzed in a different way. So I think new experiments are necessary really to look at that. And I think more accurate tracing systems where you could, maybe mutation rates in cells, they're not very high, but every cell division introduces a new mutation. If our sequencing of whole genomes of single cells reaches a coverage, it's not there yet, right? That you could really measure that. Maybe you could, do a genetic tracing experiment by just natural mutation rates in cells. But, yeah.
Okay, thank you very much.
Thank you. Thanks.